students, welcome to Biomix Hub. Do you know how plant depends upon sunlight for the proper growth and development of the plant? And what are the wavelength of sunlight being absorbed by the plant and how they are being absorbed? So for the absorption of the sunlight, the plants are having different type of photoreceptors present in it. So there are three main photoreceptors which are present, like phytochrome, cryptochrome and phototropin. So in this video, we are going to discuss about one of the photoreceptor which is known as cryptochrome. So this cryptochrome which is present, it is absorbing the blue light. Okay, and it is made up of two things. That is the chromophore, and protein. This chromophore which is present, it is having two parts. Like it is having a flavin, and a pterin. This flavin is made up of FMN and FAD. And in case of molecular biology, you have already learned about the enzyme which is known as photolysase, and this photolysase is responsible for the DNA repair mechanism. So this cryptochrome which is present, it is structurally similar to photolysase enzyme. It is structurally similar with photolysase enzyme. And this pterin molecule which is present, it is having a domain which is known as MTHF. So what is the full form of MTHF? It is methyl tetrahydrofolate. It is methyl tetrahydrofolate. Okay, so now let us discuss how the structure of this cryptochrome is present. This cryptochrome is having two domains, a N-terminal domain and a C-terminal domain. In the N-terminal domain is present PHR, next is MTHF or FAD, and in the C-terminal is the CCT domain. This is a part where the light falls, and this is also known as photosensory part, where the light is being absorbed, and the CCT is the response regulator. It is the response regulator. When the light is absorbed, there is some of the structural changes in the CCT domain and the response is being done. So in general, the cryptochromes which are present, there are three type of cryptochromes, like Cry1, Cry2 and Cry3. These are the three type of cryptochromes which are present. This Cry1 which is present, it is light stable, and the Cry2 which is present, it is light unstable. Okay, and the Cry3 which is present, its function is unknown till now. And this Cry1 and Cry2 which are present, they are present in the nucleus. So the location of these two proteins which is known as Cry1 and Cry2, it is in the nucleus. So as we are discussing here about the Cry protein or the cryptochrome, so the main work of the cryptochrome is that it will absorb the blue light from the sun, and after absorbing it, it is having the ability to inhibit the stem length. Okay, so what happens in case of that? The elongation of stem is taking place. The hypocotyl length is being increased. So for this improper elongation, there is a regulation which is being controlled by the sunlight, and it is being received through this photoreceptor which is known as cryptochrome. And upon the function of this cryptochrome, this elongation of this hypocotyl is being inhibited. So in short, we can tell that this cryptochrome has the ability to inhibit the stem. Okay, as well as it can help in the production of anthocyanin. The first work is that it will inhibit stem elongation. The other work is that it will help in accumulation of anthocyanin, as well as it will help in leaf expansion. Okay, so there was a protein which was known as HY4, and upon the mutation, this HY4 which was present, it was first of all, it was identified in a plant which is known as model plant which is known as Arabidopsis. It is known as Arabidopsis, and this HY4 which was present, this protein was mutated when it was studied, and it was observed that the length of the hypocotyl, it was elongated. Okay, later this HY4 was named as Cry protein. Okay, so now let us see how this is being regulated, how this anthocyanin accumulation and hypocotyl length is being accumulated by these cry proteins. So what we have discussed that the anthocyanin which is present and it is being regulated by the cryptochrome, and for this cryptochrome there is production of anthocyanin. So here we can see that this is cry1 which is present. If in a plant there is high amount of cry1, so during this time there is production, high level of production of anthocyanin. Here the production of anthocyanin is high. Okay, next here we will see in the wild type. What happens in the wild type? It is here the cryptochrome which is present, it is not overexpressed. So in the wild type there is little accumulation of this, there is moderate accumulation of this anthocyanin, but when it is overexpressed the production of anthocyanin is more. When there is mutation of this cry1, the production of anthocyanin is also less. Okay, now the second condition about the hypocotyl length. What we have studied that the cryptochrome which is present, it is responsible for inhibiting the hypocotyl length. So here we can see here the cry1 protein which is present, it is overexpressed. Okay, here it is overexpressed, that means cry1 protein is present over here, so it will inhibit the length of the hypocotyl. So here the hypocotyl length is small. Here we can see the wild type condition. In the wild type condition the hypocotyl length is in medium amount, because here no doubt the cry protein is present over here, so it is inhibiting. When we see the mutated cry protein, here the cry protein is not working, but the work of the cry protein was, it was inhibiting the hypocotyl length. Now what if it is not present? This cry protein, if it is not present, it will help in the elongation of hypocotyl, it will not inhibit the hypocotyl length. So here we can see here the production of hypocotyl or the length of hypocotyl is more. Okay, this is how the cryptochrome is regulating the hypocotyl length and anthocyanin pigment. Okay, next there are some of the proteins which are being regulated due to the presence of sunlight. So let us see. There are three types of proteins like SPA1, COP1 and HY5. This HY5 which is present, it is a transcription factor, and this transcription factor is responsible for photomorphogenesis. That means what you mean by photomorphogenesis? The plant will bend towards the light during the process of photomorphogenesis. In presence of light the development will take place and it will bend towards the light. So in the night condition we will see three conditions: in presence of light, in presence of sun, and when it is mutated. Okay, in the first case we will see what happens in the night. In the dark condition, what happens? In the dark condition, this cry1 which is present, it is in the inactive state and it is also having a CCT domain in its C terminal end. So the light absorbing part, the N terminal was the light absorbing part, so this N terminal is inactive, so there is no light, so there is no absorption of the light. So during this case the COP1 is present inside the nucleus, and this COP1 is binding with another protein which is known as SPA1. Okay, next the COP1 and SPA1, when they are in complex, it binds with HY5 and it ubiquitinates, polyubiquitinates the HY5, which undergo 26S proteasomal degradation. So here the HY5 which was present, which was responsible for transcription of photomorphogenetic genes, now it cannot transcribe those genes. Okay, this was the condition when it is in dark condition. Now let us see what happens in the day condition when the presence of sunlight is present. Here the sunlight is present, so from the sunlight the blue light which is present, it is being activated or it is being received, and it is received by the cry1 protein. Now this cry1 is active, it phosphorylates the CCT domain. When the CCT domain is activated, it brings the SPA1 and COP1 near it and it binds them. It will not allow the COP1 to do the this COP1 which is present, it cannot bind with the HY5 protein and it cannot ubiquitinate the HY5. Okay, so as this HY5 is now free, it will help in transcription of photomorphogenetic genes. In this way, photomorphogenesis will take place by transcribing this HY5 protein, and it will go outside after the transcription. After the transcription, it will go out into the cytoplasm for the protein production. Okay, now the next condition. Then the next condition is CCT overexpression mutant. What happens if the CCT is overexpressed? CCT is overexpressed means the C-terminal end which is present, which is the response regulator, it is phosphorylated. That means its overexpression is phosphorylated. That means anytime it can express. It does not matter that whether it is a dark condition or whether it is a dark condition or during the day. Okay, so as it is being phosphorylated here, it will catch the SPA1 and COP1 with it, and it will allow the HY5 to transcribe, due to which there will be photomorphogenesis. Here the HY5 will be produce the mRNA and it will be transported out from the nucleus where it will form the cytoplasm, where it will form the protein in the cytoplasm, and it will help in photomorphogenesis. These are the three conditions which are being regulated during the dark, during the night condition, and when the CCT domain which is present, it is overexpressed, mutant. Okay, this is how the regulation is taking place. I hope these videos are helpful for you, and if you like this video, please do not forget to like, share and subscribe. Thank you.